Good morning, everyone. So it's day 2 of a ZAPI stakeholders meeting conference that will be dedicated mostly on the discussion on findings, learnings, regulatory improvements, and discussion on the future after ZAPI with several roundtables. We'll start with session 3A that I will drive, that will be dedicated on the main findings, and we'll maybe repeat some discussions we had yesterday. I'm sharing my screen. Here. And Lekan, you know you can jump anytime and with AB on my comments. So main ZAPI findings on the topic of vaccines and the new way to design vaccines, on the topic of antibodies and other topics. Just a reminder of what were the initial ZAPI objectives, because time flies, and it was reminded yesterday several times that the concept of the ZAPI project was initiated in 2013-14, and the project started in 2015. And of course, in between, we had several improvements in technologies and also acceleration of some innovation and technologies due to the SARS-CoV-2 occurrence. And that was, in fact, something we were expecting, not so soon. So we are not completely ready, but we have very good basis for the next one. So in terms of vaccines, the objective was really to develop a monovalent vaccine to address a pandemic or a panzootic disease caused by a single virus. As of today, we see with SARS-CoV-2, possibly with variants. That's something we did not incorporate in the concept, but as you have seen, the system today is flexible enough so we can adapt. And the first idea was we wanted to design soluble subunits because it's something easy to manufacture and to provide good yields to achieve the surge manufacturing objective, more than a live virus or other things. And typically, we had some idea from the start to use particles, especially kind of multimeric protein scaffold particle, but as a normal, or I would say classical fusion between proteins at the time, because the bacterial superglue was not known when we started. And because it's always easy and more demonstrative to go into animal diseases, because you can use right away the target species, veterinary vaccines was a priority. And also a second rationale behind that was that in animal health, we are facing new diseases quite frequently, more frequently than in the human population. So every 4, 5, 6 years, we have major outbreaks in some parts of the world. And typically we had 2 strong ones in Europe in the past 10 years. The idea, of course, as you have understood, was to have a fully industrial scalable process and with a similar downstream process, both for veterinary vaccines. So, of course, downstream processing compatible in terms of steps, equipment, and cost with a safe and cheap vaccine, because that's what we need for animal health, especially production animals. But also the same with further purification, meeting regulatory requirements for human vaccines in order to have exactly the same approach for the 2 types of vaccine, but with the same design. On the antibody part, The idea was to go and to identify highly potent neutralizing antibodies against MERS and Rift, two different examples of zoonosis affecting people. And the idea initial was to design a fast-track manufacturing in order to accelerate the supply of therapeutic treatments to the patients in intensive care units. And of course, we wanted to do that as soon as possible. So in practice, it means a few months after the selection of the antibodies and the lead candidates for these antibodies. And the SARS-CoV-2 experience has shown that at least this timeline is quite realistic, because right away in March, April, we had many people to treat. The difference, and that I'm going back to my Black Swan event reference yesterday, we were thinking in a classical way that based on the previous experience, if I can say, with SARS and MERS, we would need only a few thousands of treatment over a few months. And SARS-CoV-2 proved us very wrong. So now we need to escalate that and to think at a different scale. On the modular vaccine aspect, the findings, and Martin Baer expressed that very well yesterday. He was surprised, and myself also to some extent, but relatively small soluble subunits can be designed and are still able to induce full protection against very stringent challenges in different models. And we have done that for 3 prototype models used in the ZAPI project. So establishing a good basis for expanding to other targets. A drawback, if I can say, is that, or a confirmation of previous knowledge, but we know for sure that glycosylation is needed for the immunogen subunits, even if they are small, at least for the 3 targets we have chosen. We tried to use E. coli because it's an easy and very cheap system to express subunits, but clearly when you do that, you cannot get protection. So glycosylation is a key feature, probably not for immunogenicity, more for the proper folding of your target subunit. But actually, any glycosylation can do. If you compare baculo, the S2 Drosophila cell line that we tested, the fungus C1, and other systems, you still get good protection with the same subunit, even if the glycosylation differs a bit. And it could even be beneficial. We did not explore that, but the glycosylation from the fungus can be even more immunogenic to some extent than S2 or baculo. What we have learned as well is we demonstrated is that in silico screening, especially subunit targeting fragments of a big glycoprotein bearing the key epitopes, at least today you can design and select and identify B and T epitopes. And if you have several of them, B and T grouped on the same subunit, typically it means that you have a likely good candidate for protection. At least this approach works quite well for diseases or viruses which are causing acute emerging and transboundary infections. Probably not, as we discussed yesterday, for chronic infections or targets which are still eluding our research, like African swine fever. Another interesting finding is that the flexibility of the multimeric protein scaffold particle for particle display. And I think we have been very good and thinking ahead that we could jump very early, all ZAPI partners, on what I call the bacterial superglue system bandwagon, which is, or which was initially very small curiosity in a lab, I think, but very fast, this system supported a completely new way of thinking in terms of design of proteins at large and vaccines and antibodies, as you have seen as well in our project. And based on that, we completely redesigned the modular approach that we had in mind initially. And that's part of the flexibility or maybe the success of the ZAPI project. There was enough flexibility built in from the start that we could adapt to innovative technologies coming in during the course of the project. And also, that's a good support of IMI. In fact, that IMI projects are long enough, 5 or sometimes longer, that we can incorporate this type of changes during the course of the project. But it is really a remarkable system because it provides very high flexibility in different ways. You have also another level of flexibility. That's what we discovered, and we have seen that yesterday. There is an open choice for the MPSP, so it's anyone can select its favorite MPSP. There is clearly no impact of the particle size between 20 and up to 80 nanometers. So as long as you are in this range, which is the exact range that the immune system has been trained through evolution to recognize. We have also an open choice for the superglue moieties orientation. So you can put one, let's say, spike tag on the MPSP and spike catcher on the subunit or the reverse. And you can also position the moiety, superglue moiety, either at the center or end terminus of your protein, either the monomer of the MPSP or the subunit. And of course, we did not explore all of that because as soon as we had good results, very good protection, we didn't want to revisit, but clearly that will be discussed in the learnings, something we can explore in the future with ZAPI partners or other projects. And in fact, this modular approach enables also a robust manufacturing process. So that's something which was not clearly obvious from the start. It looks more complex because you have two components to manufacture, but in fact, you reduce and you remove bottlenecks and constraints because probably many of you know, in molecular biology, when you try to fuse two different proteins together, it's a nightmare because you have to do dozens of trials and errors with different linkers to find the right sequence allowing the right folding. Here it's very simple. You just express the two separate components, the MPSP, which will be in the future the invariable component. It will be the constant basis. And then in case of emergency, you just need to express the target subunit, which is, of course, the variable component adapted to the occurring outbreak. And we have demonstrated that this coupling is quite simple to achieve. And in the next few months, we'll scale up that to confirm, but we have no worry about efficacy of this step. It is highly robust. As I said yesterday, it has been used by different people in our labs with no training, and if you respect the conditions for the mixing, it goes very nicely. And also a very good surprise and very good result, which was also not expected at this level, is that thanks to the ZAPI project, the C1 fungus system has improved a lot from the start to now. And we can say we have now very high yields, which also are driving a new way of thinking on how to manufacture MPSP, because we passed with that from a limiting bottleneck in the expression of subunit, which is typical, to a limiting now step for the expression and manufacturing of MPSP. a coupling step and the super glue. For me, it's I've been living through the innovation of PCR. That was really something fantastic when you try to do mutations. So that was when I was in US in the early 90s. So overnight you could manipulate the sequence and make any mutation you wanted very easily. And here it's a completely new way. You can play with proteins in a new way, and you don't even need to go through the DNA step. You are using this family of super glue. There are several systems which are highly specific, as specific as restriction enzymes. It's really a breakthrough technology, and we'll see how the different applications in the future will develop. So we can really go into now a smart design of multifunctional proteins and enzymatic systems, whatever. But for vaccines, again, it's a new way of thinking because it's very easy and simple to perform. It has no impact on the immunogenicity and no interference between repeated vaccinations. It's again very robust. It can be done with semi-purified components up to the use of crude culture supernatants. And that could be a way to purify MPSP complexes because you can make the complex in unpurified supernatants. And then because of the size, like antibodies we have seen yesterday, you can just use membrane for processing. It is working with a large range of molar concentrations, and you can really achieve exactly what you have designed with using this system. Another important finding is also, and not expected from the start, is that we had this objective to establish a full in vitro QC analytical system, because more and more, and that's a very strong recommendation from EU and now FDA as well and USDA, is to remove animals for the QC qualification of the batches. But designing a biophysical object as a particle decorated with a subunit opens the way to use completely new tools for assessing the quality of the vaccine and the final product. And during this approach, we and the partners and my colleagues at Hanover have been able to generate antibodies specific against the anti-superglue moieties, at least spike-tag, spike-catcher. We have not worked with another system. We have also antibodies specific for the MPSP. And especially, we have developed these biophysical assays that I presented yesterday, and that could be, in the future, the only QC assays needed. So only two types of techniques without need. I know it's kind of very provocative for regulatory people, possibly no need for specific antibodies. And that would be something really, really new for vaccines. Another comment and finding on this modular particle display based vaccines, we have high potency activity with potentially very low dose, and that of course has a direct impact on the manufacturing capacity because this is arithmetical, and that translates into the surface of your manufacturing plant and size of equipment, and of course the cost, and you know the cost of equipment is exponential with the size typically, not linear. And what is interesting so far, and we did not explore all the limits of the system. I think other teams are doing that also currently. There is no abuse differences as long as we can appreciate between saturated particles, which are bearing 50 to 60 subunits, or partially saturated, so medium saturated, 20 to 40, or what I call as weakly saturated or decorated particles, which have less than 20 subunits per MPSP. And as we have seen in all examples described yesterday, there is a clearly very positive effect on the immunogenicity of these MPSP complexes in different species as well. And that's in line, if I go back, that's in line with the general thinking and knowledge in immunology that particles are drained to the lymph nodes and then they meet immediately the right immune system. But there is also something else in the size, which are alike viruses, and probably just the immune system is better trained to react to this type of size than to other bigger size like bacteria or microparticles. Again, I will have to leave these questions to our immunologist colleagues. And to give you an idea of the potential manufacturing capacity, taking the Schmallenberg subunit system here. So if we assess that we can reach with the scale up 2 grams per liter in the supernatant in the C1 system, using a 20 microgram dose, which was our initial target, you have already 100,000 doses per liter, which is quite good. But if we can go down to 1 microgram, it's 2 million doses. And of course, that's the same. You need only 8.6 liters to do these 100K doses. And we don't see on the slide, I'm sorry, I forgot the animation. You just need to divide that, but meaning that you need less than 1 liter, half a liter. Or actually you have the same volume, but you have 20 times more doses. And for culture fermentation, also assessing here on the right, different scenario. So either a target of 1 million doses in a few months or 100 million at 2 doses. So it means that for 20 microgram, we would need 1,000 liters fermentation, which is still very accessible in industry. And that's already a huge amount. And you can do that in parallel if you want. And if you are as low as 1 microgram, it means that you have only 50 liters of fermentation of C1. And there would be, of course, a little more for the MPSP, about 4 times more with the losses. You need 200 or 250 liters of fermentation of E. coli, which is also very reachable in our conditions. And jumping to 1 billion doses, you still have figures which are manageable in a single plant or several plants. And due to the portability of the ZAPI approach, we can actually multiply several small volumes, which could be easier to handle in the future. In terms of process timelines, that will be a final deliverable, and we'll describe that in the white paper that will come in a few months. The MPSP is the invariable component, but for a process in E. coli, you need up to 3 weeks to include everything, whatever the scale. And for the C1 specific subunit construct, you have to take into account, as Ronen described, the generation and the screening of the base construct first, typically about 2 months now, maybe a little less in the future. But then you ramp up very fast because you just need 1 month more, maybe less, to reach 100 liters and then 1,000 liters. So meaning that in less than 4 months, you have the capacity to produce in a single site or 100 million doses at 20 microgram, and of course, much more if it's a lower dose. So probably one of the key findings in our capacity to react and to be prepared against the next big one. And on top of that, of course, you have to add the other steps, because the manufacturing is not only the manufacturing of raw material components. You need the coupling step, and typically this will be overnight. So not adding a day in the system. And there is no impact on the overall manufacturing cycle time. And the QC assays, we are, of course, these are experimental by nature today, but we think that we can complete them in a few days once we have the equipment on site. The A4F is very rapid in terms of analysis. You just inject the sample and you get the data in a few minutes or a few hours to analyze, and it will be the same for mass spectrometry. Actually, the longest test will still be the sterility test, because as per pharmacopoeia, you need at least 2 weeks to give this result. And there are systems to accelerate that, but they are not GMP. So the GMP test is still 2 to 3 weeks. And that's, in fact, strangely enough, now a bottleneck. But of course, after that, and we can deduce that from the information which is disseminated right now about the supply of COVID-19 vaccines. Many bottlenecks can occur after the manufacturing or at the end. You need to ensure that you have a supply of adjuvant large enough to have all your filling lines capacity working and no failure, because they break quite often, enough vials, enough stoppers, and so on. But again, the learnings of the SARS-CoV-2 crisis will, I hope, will help us to be better prepared. And to go into the QC in vitro assays and 3R, so compliance. In fact, again, to be very provocative, you can think that we need only the two biophysical assays, A4F and mass spec, to get all the information we need on the identity of the MPSP and subunit, the coupling ratio, and to assess it directly by mass spec or by the molecular weight through the A4F system and the average diameter size. In terms of antibodies, finding is not really my specialty, and so Lekan, please jump in if I say something wrong. You have where we can probably improve is the selection and screening of antibodies that took actually a relatively long time in the ZAPI project, except for the HH nanobodies, which are maybe the fastest way to generate or to identify good antibodies. But if you go to immunization, you need to ensure that you have success in immunization, and there are failures. You don't have always the right combination between the target species or the transgenic mice and your antigen. And then you need to screen for neutralization and protection. But of course, it takes some time. But once you have the lead candidate, you can go fast in manufacturing. Finding, and now it has been also used and demonstrated by other teams in the world, is that coupling of VHH with the superglue is a very promising approach to develop potent new neutralizing multi-specific antibodies, because you generate the neutralizing activities that the single VHH nanobodies don't have. And that's also quite surprising. And it's a new way of thinking for the future to screen new antibodies or new types of antibodies. Of course, the structural and functional profiling of antibodies can also aid in vaccine design, because you consolidate the information you have if you are using the neutralizing antibodies to confirm that the subunits that you have selected is also the right one. Here also on the antibody isolation strategies, we have understood that you need to align with the new rapid manufacturing strategy. So it doesn't make sense if you are able to manufacture an antibody in 2 months, if it takes 2 years to select it. And today, again, with the SARS-CoV-2, we have seen that several teams have been able to clone very fast from patients very good neutralizing antibodies, so directly from the human B cells. And that can be applicable to other targets and also in the animal health field. The learnings and findings that all the previous knowledge on in silico sequence analysis and developability, manufacturability, they are indeed important, but not completely predictive, as we have seen. And we still have to learn in this area. And as demonstrated very clearly by Lekan and his team yesterday, the transient expression platform can clearly be used to generate high quality antibodies, but to some limitation in terms of amount. And clearly also, thanks to the ZAPI project, strategies for purification analytical have been developed and improved in terms to meet the fast track requirements. And I think that's also an important achievement. So it's about the same message that product quality consistency has been demonstrated. Also using transient expression platform, we cannot really distinguish with a clonal established cell line. They can also accelerate the validation of a lead antibody to be first in human and at least to treat the first patients, but also to indeed to confirm that you have a right candidate, and then you put it in the clonal system. And of course, again, that was maybe our mistake, if I can say, quote unquote, because nobody could think of the scale of the outbreak that we have today, but it's a fact. So now we have to consider this as a fact and as a target. The suitability of the transient expression is limited. It depends on the number of doses and the patient number. So it's good for one country and a limited outbreak, not for a global outbreak. But it's a first step that we can take anyway.
I don't know if I can just quickly add to that. Really, I mean, you covered you know, most of it. I was just going to add that with transients, obviously the bottleneck is DNA. So with everything, I mean, in terms of rapid response, is preparation as well. So it would mean having a facility or a means of quickly making plasmid DNA for it, because that would also drive, you know, increase the speed. The other point made yesterday, which is true, is in terms of cost. Obviously, the RNA platform, it's not the cheapest as well for the COVID, if you could, but it's because of the pre-work that they were able to do before that allowed it to go faster. So it's not necessarily going to be the cheapest, but it can be made faster. Thanks.
Okay, that's something I'm addressing also in the next session on the learnings about the DNA. So we have just a few slides on regulatory innovation as well. Joris or Carmen, if you want to comment on these slides.
Yeah, short comments. We had, when we started with ZAPI, there was for sure that we had no legal basis for quick licensing, emergency licensing of modern products. And so we tried to give some proposals, provide some proposals, also based on the lessons learned. What is from the scientific point possible, from the manufacturer's point possible, to go ahead in these situations, and therefore we initiated the proposal for a platform master file. I will say something about this afternoon. We were very lucky that we had the option or possibility to include some of our proposals in the new regulation. This was also the good interaction between Joris van de Putte and Madame Grostet. And we made some proposals as well. And I'm very happy that EMA was taking our proposals on board and is supporting these approaches. What helps as well now is this COVID-19 situation. For now, all regulators throughout the world have learned that we have to change our approach in licensing. And okay, that's all. And ZAPI can contribute and is somewhere in the middle of the whole new development. And this is a very good situation.
Yes, and it was indeed a key achievement to have this platform master file now implemented, included in the new regulation, or at least on veterinary medicinal products.
Well, as Ivo said yesterday, it's a proposal, it goes to public consultation, and it follows the whole process. So it's not final yet, but personally, I think we are on the good way, but we should not preclude the public consultation and then the whole process with the European Commission.
What we have done is we made the first steps, and if you do not start walking, you will never reach your goal. And it's so far. Even if the way will be longer, then it doesn't matter. We are walking.
But I think we can say that what we achieved so far was unexpected when we started.
Yes, I agree.
Jean-Christophe, shall I chip in some slides which also introduce the next 3B?
We are still on 3A, and
I know,
but you want to
binding
I think as probably the last one on regulatory that we just commented. In fact, yes, you can. You can share your slide.
Yeah, perhaps as a prelude to also the next session, the 3B, and let me try to share the screen.
I have an eye on the time, but it should be okay. The 3B is maybe shorter.
I think so. Yeah. So can you see my screen?
Yes, yes.
Okay, so I made some slides, and it's also a prelude to what we're going to conclude at the end of the day. And this is about what was the origin of the project and how is it going to help us in the future. And if you look at this slide here, what you clearly see is that since the, let's say, 1950s, or from the 1950s onwards, we had this more generic complacency that infectious diseases were under control in the Western world. And then when I entered the field, shortly thereafter, we got HIV, and we got an enormous increase of all these different infections. And we were facing even 3 pandemics, the last one we are still in today. And these are pandemics. Of course, in the veterinary field, I have a similar thing, which I may be presenting later today, for the animal diseases, but that's even much more serious. And it's really the way in which we currently dealing with the planet and all the changes that are going on. So this was the reason that we really thought about the necessity of a project like the ZAPI project. And this was not too long after the last pandemic. And if you look here at this slide, yeah, so on the left-hand side, you see the real life situation. And it took really too long to make a vaccine, because at the southern hemisphere, we had the peak, and just after the peak, we had the vaccine. And I was involved in flu block, became involved in flu block as well. And then we realized with the technology that we had, you could basically within 3 months, yeah, you could really reach a considerable vaccine production that came to late here. And so the idea was really, what can we do? What are the new technologies that we might be able to use for the future? And so I think the ZAPI has contributed tremendously to being prepared for future outbreaks. And as Jean-Christophe said rightly, we started 5 years ago. And if we would have started a couple of years earlier, yeah, perhaps not having all the technologies, but then we might have come up with a vaccine even faster, yeah, and with antibodies. So if you look at interventions that are possible, yeah, so it's identification of correlates of protection. That's the first thing, yeah. And then what can we do? We can make protective antibodies. We can go for protective antibody responses by the right vaccines, and therefore you have to identify the proper antibody targets for neutralizing antibodies. And yesterday, very briefly, we also talked about ADCC and antibodies with other functions. What we have not touched upon, and really be focused on the first part, is protective T-cell responses. And again, broadly reactive. And that's also true for the antibody responses as we have seen yesterday. So, and that's indeed identification of the right T-cell targets, yeah, CD4s, CD8s, and the different subsets. I think it's important to realize that for future vaccines, yeah, we need to have this knowledge. If you look back, obviously, already 2 centuries ago, a vaccine was made against smallpox without knowing anything even about antibodies and T cells. But to date, we are in a position that we can really target our efforts into this direction. What we have not talked about, and that is still a thing that might come up during this pandemic, is correlates of immune pathogenesis. So not immune protection, but immune pathogenesis. And this can be, on the one hand, antibody-mediated enhancement, as we have clearly seen in for instance, in the coronavirus infection of the cat, where classical vaccines actually predisposed for more serious disease, classically inactivated vaccine. And the other thing is T-cell mediated hypersensitivity, as we have seen, for instance, with RSV, yeah, but also with other infection, even with the coronavirus infection in monkey experiments. So I think this is an important thing to realize. We have touched upon a very important thing, and I think the strength of the project was really the focus that we had. I'm not going into this slide because it summarizes really what we have done, and we'll come back in the overview later today. Yeah, so selection of the immunogens on the one hand, production of the key immunogens, in vitro characterization. This is all related to vaccines, and a similar thing went on for antibodies. So I think this is quite important to realize that we have been doing that quite efficiently. Well, if we now look at what we are throwing at vaccines for SARS-CoV-2, and you just look at the right-hand side at the top here. Yeah, so we have about 80 different vaccines that are being tried. Yeah, a number that have been in preclinical and clinical trials have been abandoned already. But you see, there's an enormous worldwide effort to do that. And for those who are really interested, you can follow it on a day-to-day basis in the Corona Vaccine Tracker in the New York Times, because it gives you all the information you need on a daily basis. Well, where do we fit into this? So we have been focusing predominantly on neutralizing antibodies, and rightly so. Yeah, and of course I talked about cytotoxic T cell lymphocytes, T lymphocytes, there's T helper cells, of course. So there is a lot about that. And where does our project impact on this, on all these things? And I think when we talk about mRNA vaccines that are so successful to date, really identifying those stretches of RNA that should be used in the vaccine development. Yeah, they really have been identified, and they will be identified by this technology. And then for the recombinant viral factors, it's basically a similar thing. We go for the glycoprotein as long as we go for the antibody responses, at least, but perhaps also for the T helper responses. And then the discussion, as we had it yesterday, are we going just for the receptor binding domain or for the S1 or the whole spike? So again, it's very important. And then of course, we have the recombinant subunit vaccines plus or minus adjuvants, the Novavax approach, yeah, where they use an adjuvant with subunits. And I can say we at the moment are quite active in this area as well. We can be active, and it would have been nice if we would have had this a little bit earlier, because then we would have a vaccine ready, I think. Well, basically, I showed you this slide yesterday, and that's the work that was carried out by Bart Haagmans and Berend Jan. In that, those are the studies that were carried out in mice, yeah, where indeed the C1 expression system was being used, yeah, and then expressing the RBD, yeah, the receptor binding domain of the virus, and you get very good neutralizing antibodies. And this has led now to a systematic study that we are carrying out together in hamsters, where we really look at the different antigen presentation forms, what is the best. And we had a lengthy discussion yesterday, which was quite important, I think, about the use of high antigen doses, the use of adjuvants, the use of the scaffolds, and all these things being tested against each other. This is ongoing, and I think for the major advantage of the C1 system, I think that they have alluded to yesterday, is the fact that you can produce enormous quantities at a relatively low cost. So what we are going to face, obviously, in this particular pandemic, is at the end of the day, if you want to get rid of the big problems, we also have to vaccinate people in middle and low income countries. So I think this will be quite an important spin-off of the work we have been doing. If we then move on, yeah. So basically, my conclusion of this whole story, and I will be coming back to that later today, is what we would have to do is in the future, indeed, invest in more ZAPI-like projects in peacetime. So for the development of broadly reactive vaccines and antibodies, and the same holds true, obviously, for antivirals and possibly also for biological response modifiers. I think the major thing here is that we have to do that in peacetime. And it's quite important to realize that to date we are in the middle of a crisis, yeah. So we are relatively late with the development of the vaccines, although it goes very fast. But I think we can do better. But if we have broadly reactive antibodies and vaccines, one of the examples is the work that Berend and Bart were doing with the antibodies, having an antibody against the MERS coronavirus that was broadly reactive. So if we will be having these kind of antibodies and vaccines in the future, and also these antivirals, we would be much better prepared for the beginning of an outbreak, and we could possibly even nib it in the bud. So I think what we would have to do is we would really have to invest in initiatives that will target conserve targets of virus families like the coronaviruses, but there is a whole list of viruses, we'll come back to that later, or virus groups within the families, if that would be too ambitious. And I think this could really be seen as a major cost-effective pandemic insurance policy. You can say this project was very expensive, of course, for the European Commission. But if you see what has been achieved, and if we were to do more of these projects against not only the low-hanging fruit, but also the other families, I think we would be better prepared. And I think if we are talking about, if we talk about a project like this, it has cost many millions of euros. I realize that, and it has yielded a lot. But if you see what the problems are with an outbreak in the animal world, a pandemic in the animal world, an animal pandemic or a human pandemic, I think the costs of those, as we see today, we talking billions, not billions, but tens of billions per country even. So I think that the technology is progressing so fast that indeed we should see it as a major cost-effective pandemic insurance policy. I think this is my major lesson for what we have been doing today. So these are some more generic remarks I wanted to make, Jean-Christophe, because I think we are all applauding what we have been doing, and rightly so. We have done a fantastic job, and I think this should be a guide also for the Commission and the international community to see that this is the way we need to go in the future. Single out virus families, and already, I'll come back to that later, already WHO in its blueprint for research priorities has already identified the virus families, virus groups that should be in the focus of our efforts. So I'll come back to these more general points later today, but I think it's quite important to realize that we are applauding ourselves for the technologies that have been developed. It's fantastic, but we should invest at least 10 to 50 times more, in 10 to 50 times more of these kind of projects.
Thank you, Ab. We are quite over time, but it was interesting to hear that, and the 3B actually will be shorter in terms of session and reviewing the same ideas from a different angle. So we should now have a Q&A session coordinated by Jean-Marie, and we may have some questions already.
Yeah, effectively, we have on the chat box 3 questions at the moment. So the first question from Catherine Charrer is, what are the conditions of conservation and shipping of those vaccine candidates? Can they be handled at room temperature? This is the first question. And in the second and third can be treated in the same time. What could be done to assess the potential for the immune enhancement? Do we have tests that can be run to evaluate this? And the third question is to Ab. The cost of this project is not a cost, but an investment. So who want to start to?
Well, the last one, I can be very fast. That's the one by yours. The cost in this project is not a cost, but an investment. And I think he has got the message indeed. Yeah. So basically, we should see this as an investment in the future.
Yeah, and I will even emphasize that it's not a big cost. And Pierre Melien reminded that it is actually one of the smallest projects in the IMI program. So we have 20 millions and actually 10 millions from the Commission and 10 millions provided by in-kind commitment from the industry. It's a very small project compared to the big ones on antimicrobial resistance, which are reaching 200 millions. And indeed, it's not only an investment, it's insurance for the future. And more preparing vaccines for future outbreaks has to be considered as a global insurance. And industry will not pay for that. We have to find a new system to provide the resources on that, and probably ZAPI is a way to go, and we may need both, I think, a EU BARDA at least, and then a ZAPI for animal vaccines as well. I will go on the so conservation and shipping. That's ongoing because we need to prepare a sufficient amount of vaccines to perform the accelerated stability studies. There are two parts to consider: the MPSP complex, which should be stable based on the natural feature of the MPSP itself. It's very sturdy and will go at room temperature or even. And then the adjuvant, and the adjuvant, at least the commercial adjuvants we have for animal health, are used at or stored at 4 degrees, but we can be shipped and used at room temperature. And probably we can ship the two components differently, but the conclusion is that we expect to have a vaccine that can be or that will be handled in the not normal conditions as other vaccines. Hopefully at room temperature if we go this way. But we'll know in a few months, not today.
Okay.
Can I take the second one, which the question, what could be done to assess the potential for immune enhancement? Do we have tests that can be used to evaluate this? I think this is quite an important point. I mentioned that for coronaviruses, we know that for the feline coronavirus that causes FIP, feline infectious peritonitis, if you use a whole inactivated vaccine, that the cats are more sensitive. And so there is an issue there. We know that in some of the early monkey experiments that we have done this SARS-CoV-1 with the first SARS coronavirus, that we saw that with a similar approach, there was a tendency of these animals getting hypersensitivity, eosinophilia in the lungs. So we are, and that's a follow-up of this project as well, involved in another big European project, which is called CARE. And in that particular project, what we are doing is that we are studying the potential in vitro for enhancement. So that means we look at immunized animals, and we look at antibody profiles, and then we have several in vitro tests. Then, of course, you can study that. If you talk about a cat, it's easy. You can do a cat experiment. I think there's been a lengthy discussion about the use of non-human primates for this particular purpose. It has been stated by the regulatory authorities that we do not need to have these evaluations in monkeys before we start using in the clinic the antibodies and the vaccines. Well, so far so good. In the meantime, however, projects have been granted. I mentioned one that we are involved in, but there's a couple of other ones where very specifically in non-human primate models, we are going to look into the possibility of these things emerging after vaccination or antibody therapy. So the non-human primate is mentioned there because the immune system and also the antibody production in rodents, and when we are doing most of the experiments in hamsters, obviously the enhancement study will be done in hamsters as well. But at the end of the day, I think there is a need to do these final experiments in macaques, in non-human primates as well. Obviously, this is an issue of discussion. We are gearing up for those experiments with the antibodies we have and also the candidate vaccines that we have. Over.
Okay, so we don't have more questions, but we have a comment on the chat box. For veterinary vaccine, the concept paper vaccine platform master file is out for consultation on the EMA website, and this is the link. And is there any other question from the audience on this different topic today?
Yes.
Yeah, I see Jeff. Yeah, Jeff. Yeah.
I just wanted to pick up on Abe's point and the CARE program that he mentioned, because it's something that dogs the whole of vaccinology. When you're developing new vaccines, the concern about enhancement. And I'm really interested to know what the CARE program does about this. In fact, I think my view is we worry about it a bit too much. I mean, we've seen it in RSV. We saw it with atypical measles, with killed measles vaccines. But otherwise, it's not common. You have something going on with dengue, but that's more of a sort of serotype mismatch problem. And then, of course, there is the example that I've mentioned of the feline corona. It seems to me, Abe, that there ought to be ways to get at this in a more scientifically rigorous manner. I don't know what the underlying causes are, but I've always felt that it's probably to do with tropism of the virus and the ability of the virus to replicate in cells whose job it is to clear out the virus, like macrophage and polymorph and nucleocytes and so on, that take up virus via an FC receptor interaction after the binding of antibody. But I think as we move forward, it would be good to have some sort of tests for this sort of thing that we can rely on. And the most things that I've seen are not really indicating whether you've got enhancement. I just encouraging Abe to tell us a little more on what he has in mind for the program.
Yeah, thank you for that. So you're quite right in saying that let's say the approaches so far have not been too scientific overall. Perhaps it's good to make a distinction between, on the one hand, the antibody-mediated enhancement, which is not a trivial thing, because you rightly said that this is probably related to mismatched strains. So we know that for dengue, if you have been pre-infected with one of the dengue virus strains and you get infected by the second, you're much more prone to develop serious disease, and that seems to be true. And we have seen the consequences of that recently. That at least that can give a major problems. One of the issues there is, I think we should keep in mind that when we are talking about this coronavirus, and coronavirus in different animal species, they have a tendency to develop many strains. If we just look at IBV, infectious bronchitis virus in chickens, but there are, if you look at the human coronaviruses, so we have been doing quite a lot of work in the meantime on the T-cell responses against the different coronaviruses and the cross-reactive T-cell responses that do exist, and that's quite an important thing. But coming back to the antibody story, we don't know whether antibodies against one of the related coronaviruses, the coronavirus like OC43, whether antibodies against those might provoke some of that reaction. We don't know, and we need to study that. And we can do that, obviously. For the antibodies, it is relatively simple because you can learn a lot from in vitro assays, from the rodent data. But at the end of the day, you should look into the sera that you have from humans and monkeys alike if you really want to do challenge experiments. So that's the antibody story. The T-cell story, you rightly said that for measles, RSV, these are quite interesting stories where we and others have shown that this is related to an aberrant T-cell response. So if you take for instance, if you use an inactivated whole measles vaccine, you vaccinate monkeys against that, and you challenge them later, and the timing is very important there. You challenge them later, you see that you could already predict on the basis of the distribution of the T-cell responses which animals are going to have problems and which animals are not. And even in those experiments, it happened to us. That when animals were challenged that had been pre-exposed to inactivated vaccine, primates that were challenged, they died from an acute shock. So as a matter of fact, I think that is quite an important thing, and not all the assays are ready today. But I think it's quite important to look at correlates of immune pathogenesis. And I understand, my worry is that when we look at the situation with both RSV and measles, as you may remember at your age, so that in the 60s, those experiments were being carried out. And so these were basically vaccines introduced in children, and as a matter of fact, the first year it was perfect; they were protected. But the balance then between the immunity on the one hand and the immune pathogenesis, with the waning of the immune response, and that was the antibody response at that time. We're not quite sure that was really the fact. We think it's much more likely that this had been related to T-cell responses. And with the waning of that, 2, 3, 4 years later, we saw the very severe cases of either measles or RSV. So it's just a word of caution. Yeah. So I realize it's very unpopular to talk today about those kind of effects, but I think it is a real thing, and let's hope we are just worried for nothing, and that that the storm will go over, but I think we have to look in, let's say, 2, 3, 4 years, what's going to happen at the end of the day.
Good.
Thank you, Jean-Marie. I think we can take the last question and then we'll have a small break.
So the last question from Catherine Charer is what do regulatory people think of QC tests not targeting specifically the antigen itself, but MPSP or superglue components? This is maybe for Carmen.
No,
No.
but I think yes, we are on the way to change the approach of the quality control of the final product. This is the introduction of the consistency principle and. But the amount of testing of a final product really depends on a case-by-case decision. And it depends on the standardization of the production, of the quality of the in-process testing, etc., etc. And therefore, I think there is no general option to say yes or no. But this has really to be discussed at the licensing procedure, case by case with the authorities. I think Joris will say something. He raises hands in some minutes.
Yes, I wanted to come back on what Ab very rightly said. This should be this kind of cost, but investment should be multiplied by 50 or 100. This can come back later on other discussions. I want to remember that at the beginning of the SARS-CoV-2 in February, remember the 65th anniversary scientific meeting in Lyon, where our American colleagues already presented their whole development and their preliminary results, and then the support of the American authorities, rightly $10.8 billion immediately on 5 projects. So what I wanted to say here is this should be an example for our European BARDA project, where a project like ours, and there will surely be other ones, but at least that they can be taken up, de-risked, and be developed in order to be able to be prepared for pandemics, zoonotics, and for the sake of public health. That's all what I wanted to say. Thank you.
Okay, so the Q&A session is over now. Thank you very much for your contribution.
Yes, I think we can start again on 3B session if everyone is back. I will share my slides. So it will be shorter, and Ab also has some slides to share after my presentation.
No, Jean-Christophe, I already shared my slides as an introduction to the
okay,
subject.
so we will save time. And
you can
but you can, you will
jump in if necessary.
Yeah, so it's a different idea. So, so we are now in the learnings and actually open questions. So it's a different view on the findings. And so Ab will help me to comment, and the floor is also open to the ZAPI work package leaders that presented yesterday if they want to comment. I think we may have an interesting Q&A session after that. So the first part that was already discussed by Ab on some aspects is the question of correlates of protection, especially with the modular particle display ZAPI-based vaccines. I think that's more a gut feeling and to be confirmed with further experiments, of course. But we focus the immune response on the key immunogenic part, and in this way, there is certainly a better correlation between the neutralizing antibodies, because what we target are bearing the key neutralizing epitopes. So when you measure these antibodies in the sera of animals, at least, you really measure the functional part and not the cellular response that can come from other ingredients or debris in the vaccine manufacturing from a typical live virus or inactivated one. So in this way, it is a way to avoid the decoy epitopes and potentially to achieve a better immune response. I'm also have a question about the quality of cellular immunity. I'm not aware of any pure antibody or pure cellular response when you immunize an animal or an individual with a vaccine. It's always a balance. So of course the balance is more on the antibody or more on the cellular, depending on the way you present the antigen, the quality of antigen, the type of adjuvant. Many parameters we don't really control and we just start to understand. But I wouldn't say today that a particle-based vaccine is not inducing cellular immunity and especially is not inducing immune memory. Because the objective of ZAPI was essentially on providing massive or tools for massive protection, so massive immunization for a short term to fight against an outbreak. And the objective is in fact to break the outbreak, as we'll see in the discussion, in the final discussion. We don't contemplate this aspect of long-term immune memory, but that's something also of interest to explore in the future, and that's open to your comments and questions. And also because of a specific aspect of this particle display system, we certainly activate both innate immunity and adaptive immunity due to a particle effect. If you remember again yesterday from Paul Wischger's presentation, and we have other data in this way with the Mrna system, just mixing the particle and the subunit has a very positive effect compared to the standalone subunit. And that's probably linked either as an adjuvant effect or because you trigger the innate immunity due to the presence of particles in the 20-80 nanometer size range on the immune system. And that in itself could be a very beneficial activity. There is also the possibility, due to the flexibility of the system, to combine your target immunogen with known strong immunogens, like fragmency of tetanus, for instance, which is a universal antigen triggering very good responses in all species. And you may end up with also a better adjuvant effect by co-presentation of different immunogens. So a lot of possibilities now are open using our system. Other open questions are and still not solved because we are still exploring that. Is there a need for 2 administrations or only 1? And of course, it depends on the type of challenge. In animal health world, we are very used. In fact, it's a process. We go directly in the target species with extremely stringent challenges, which are way above of what the animals are exposed in the nature. Typically 10, 100 times more than what we would be exposed to. And meaning that we develop very, very solid vaccines. But in the field, in a natural infection, natural outbreak from contamination and contact, maybe only 1 administration is enough. And we have seen that in Schmallenberg, at least one administration provides a sustained level of antibodies, relatively low, but quite detectable, and it should be protective for a long time. The other question is the need or not of an adjuvant. We don't know yet. It depends also on the type of adjuvant. Again, we discussed yesterday the balance that we are facing if we go without adjuvant, because that means a need to increase the payload, and that could be detrimental for the manufacturing capacity. Another question is, what is the best adjuvant for particles? And from previous knowledge also in the vaccine world, and especially animal health vaccines, we know that all types of adjuvant are not fitting well with particles or VLPs. We still don't understand what's going on, but we just observe that you have very good combination and some combinations which are not active. And on top of that, the combination, a very good combination, may work only in one species and not in another one. So it's again a complex equation when it comes to vaccinology, which is not an exact science still as of today, but we have good bricks to play with now. Another also learning, and that was not so expected, in fact, is that there is no obvious difference, at least in the models we have used, between the saturated, the partially decorated, and weakly decorated. And what does it mean? It is just the particle effect. So if you imagine that you have only one single subunit coupled to a particle, maybe it can do the job, and that's something to evaluate in the future as well. I said, so we have clearly a general positive effect of the presence of particles, and this is due to the fact that particles also are drained very fast and very efficiently to lymph nodes, and that's where the immune response is initiated. When you refer to historical experiments, when you inject China ink in the skin, or just a few minutes or seconds, 30 seconds after, your lymph node, which is the closest, is completely black, and it shows the speed of the biological reaction in the body. And playing with the particle display and the superglue capacity opens also a new possibilities to co-present immunogen subunits from different pathogens, so variants, as you can see in the paper to be published in Science. It is available as a first release with all variants and all different beta coronaviruses, with very good effect in terms of inducing a broad reactive immune response. But you can do also that with serotypes in blue tongue virus, if you can imagine this approach, and also possibly with different pathogens. So it would be a different way to design and manufacture combination vaccines. Maybe not so easy because you have still to play with the different manufacturing lines for each subunit, but eventually you could have a very stable product and it's easy to QC. Correlates of protection, and probably Ab will also discuss on that, but in the context of therapeutic antibodies, and the main learning is, yeah, we need to be better collectively to fast screen and select neutralizing antibodies. I was expecting in the call for project to have proposals around the direct cloning of antibodies, but it was not the case. So we also, and that's a learning in the IMI project, you have to use what the partners are bringing, of course, and you never have an optimal or ideal project, but I think we were very close to that with ZAPI. We discussed yesterday the question on how to screen and select the non-neutralizing in vitro antibodies, but which are very efficient in vivo, and that's an issue in itself. Because you cannot really assess outside of the animal the quality of these antibodies. But you can learn, and then you can select at least antibodies targeting specific parts of your pathogen. Discussing also the better potential of combination antibodies for therapeutic activity is also opening another question on how to screen and select the best combination, but it can be a long process. And something I wanted to comment yesterday, and that Jeroen and Paul have observed with Arbor Biomed, is that the initial experiments of immunizing mice with subunits from Rift were not so successful and induced a delay. But when the ZAPI-based vaccines, so particles decorated with Rift domain, were administered to mice, then there was an extremely good response in terms of neutralizing antibodies, which were not screened immediately because we came too late. But it shows also the link and the parallel between the two sides of the ZAPI project, so antibodies and vaccines, and they are complementary. So each one can help the other part. And in fact, going in the future for very efficient immunization and very focused immunization, using a particle display is probably a way to go. In terms of surge manufacturing capacity for antibodies, and Lekan, you can comment as well, we had a clear demonstration that the transient transfection is quite efficient. And as we just discussed earlier, there is a limit, and especially for the cGMP plasmid DNA supply. You need to be aware of that and to have all the equipment in place. Over than that, the scale-up can go beyond the 200 liters that were evaluated by AstraZeneca. But it also shows that facing something like COVID-19 today, you may need in the future to implement immediately in parallel 2 or 3 strategies, so the TTE, the pools of CHOs, and the clonal selection. But there is also something we did not address in the project because that was not foreseen from the start. The capacity also of a C1 fungus to express antibodies at very high level, and I think they are in the range of 20 grams per liter or more today. And also the possibility to use coupled nanobodies that could be expressed in E. coli. So maybe with possibly a shorter half-life, but again, I remind you the objective was still to treat patients in an intensive care unit. So it's a life-saving treatment. So you don't have the same requirements, same parameters. And again, a one shot or a single perfusion can do the job to block the virus in the body and then to help the patient to recover. So it's also a new way of thinking on how to use biologicals and monoclonal antibodies. Up to now, they have been used mainly for chronic disease, metabolic diseases, but fighting infectious disease, that's another approach for me. On the side of vaccines and surge manufacturing, again, the big learning is that manufacturing platform is very flexible, more flexible than what we were thinking initially. It becomes also almost an open source platform because you have a choice of a MPSP. Of course, you will have your way to design the subunit. The only proprietary system in the platform is the superglue system, but probably an easy license to acquire. And an aspect that was not discussed yesterday is the portability. So the possibility to multiply the manufacturing sites, because the process for manufacturing each component is very simple and can be transferred easily from site to site. And that will enable a global supply immediately compared to the current situation we are also observing. And the last point on this manufacturing slide is that, as I said before also, the C1 yields are creating the need to rethink now how to manufacture MPSP. Sorry, I made a typo. It was too early this morning for me. The E. coli expression we can still improve, but and stockpile in advance. But stockpiling means funding. Who will funding that? Open question as well. And there are possible more efficient systems to evaluate, improve strengths in E. coli, new bacterial systems, secreting, and C1, which is secreting, and but we need to evaluate. And possibly, maybe in a few years, we'll have a very good system fulfilling all these requirements. And again, and the question was addressed just before, this new QC approach for particle display-based vaccines. It's in line with the vaccine by design approach, and you know that in pharma industry, the quality by design is implemented more and more. I think it comes to human vaccines as well, but it would be completely new for veterinary vaccines. But the fact that we have a biophysical object, a particle that we can describe very precisely, enables that. And again, it's a quite provocative thinking, but it would allow, on paper, a reagent-free QC system. So we need now to consolidate this data. A lot of data generated by the ZAPI. We showed only the best ones, but we have many more data. It's a time over 2 days. We cannot present and discuss everything that we have done in the past 5 years. But the applicability and AB, we'll discuss that in the next session, is certainly there and quite exciting to follow in the next few years. And I will finish with on learning on the network consortium, the quality. For me, it's as it was mentioned by Pierre Melien, and in my own experience, typically the EU projects are quite complex and difficult to go with, and they don't achieve all the expected deliverables. So it's really unique to have everything working in from the start in this group, because we maybe an exceptional team of people and flexibility. We could create also a very high level of interconnection and open exchanges, and that was also a factor of success. Very high trust between all partners, very smart people, listening, understanding, daring to take new ways. It was said several times that yeah, the scientists were surprised by the results. So we pushed for innovation, so we were successful. I think it's always a matter of luck, but we are building luck on the past experience, and I think putting together industry people and scientists, academic people is the best way to go for creating these new tools that will be helpful for the humanity. And all partners clearly align on the same overall objectives for the vaccines, for antibodies. And we achieve that. And at the same time, we have done excellent science, as you have seen yesterday in some papers and in the future papers to be published in the next few months. So thank you to all the team in ZAPI and supporting team at Phinovatis and Artemis, because it's always extremely complex, and but a great experience and adventure. So Ab, your turn.
Yeah, thank you. I basically made my points before. I think there's quite a few points for discussion that have been left, and well, one of the things is that we are talking about the transition between the animal world and the human world here in the One Health spirit, yes. And that the vaccine development for animals, yeah, basically maybe the basis also because it goes faster to some extent. Maybe the basis for vaccine development for humans as well. What we have not addressed is a very fundamental point, is that in animals, of course, you have quite some differences there. On the one hand, you have the production animals, and when we are talking about chickens and pigs, or cattle. Yeah, so it's a different thing from companion animals. So the criteria for companion animals, they are more going into the direction of the human criteria, although of course we have to keep in mind, and Carmen will be stressing that undoubtedly, yeah, that there are animals that are used for food production and that may create an extra risk. But I think when we are talking about induction of protection, protective immunity, I think it's important to realize what is our target species there, and there are quite big differences there. If you look at the lifespan of a pig, yeah, or a production pig or a chicken, usually that's relatively much shorter than the human lifespan, and that's also true for companion animals. And that also means that for vaccination, you may have quite some different goals there. And obviously, we didn't address those points in this particular project, and you've quite nicely shown, Jean-Christophe, what all the benefits are. I think this issue of lifespan is quite an important one. The other thing that we did not discuss at all at the moment is the use, and it's a point that comes up in the whole discussion about SARS-CoV-2 as well. That basically the different approaches that are being chosen, they may be complementary even, yeah. So you could have strategies where you have a prime boost, yeah, and prime boost strategies. In the veterinary world, we don't like that because it causes a lot of problems, obviously. Also in the human world, but if certain people are really quite in favor, and where you see prime boost strategies popping up, it's usually in the fields that are hopeless, yeah, like HIV, malaria, and the like. Yeah, so the latter is not hopeless, I think. But for HIV, a lot of these prime boost concepts have been worked out. We have been scratching the surface, yeah, and there is a lot of things that we still have to learn. The other big problem we are facing, obviously, in humans, we know a lot about the immune system, and that's also true for mice. And so many vaccines have been made for mice that never made it into animals or humans. I think that is one of the critical issues there. Really, I think the beauty of this project was indeed that the goal was really to come up with technology, a technology or technologies to have something that can be used in the target species, being certain animal species or humans. And we have seen quite nicely with the Rift Valley fever, with the SBV, but also with the coronaviruses, that has been quite successful and has been worked out. So I think that's quite important, to take it from the stage where you go into mice and then go over to the next project that you like. Whereas this project is really, and that's in the spirit of IMI, to really take it from let's say the drawing board initially, and then go into the different animal species and the laboratory animals, but really take it to the target species. And I think that's the beauty of this project, and that's also because it's a 50% scientific, 50% industrial funding, and I think that was quite important. So there is a lot of questions that will be remaining also for this afternoon. I think the whole issue about regulatory, that is very well covered by Joris and Carmen's group. I think that is quite important as well. But also, I think what is quite important, and we will be discussing that later today, how can we use these findings, not only to come to tangible vaccines, but also how can we use the module that has been created in the ZAPI project? How can we translate that into future funding projects? And that in Europe, we create a system similar to what in the USA is the BARDA system, and there are other mechanisms there. And I think the other thing to realize is that obviously, and we have also seen that during the coronavirus crisis, that this interaction between on the one hand, industry and basic science, but on the other hand, also the geographical transcontinental collaborations, that they are quite important as well, and that funding authorities, and we see a good example there is, for instance, the ZAPI example, where indeed projects are being pushed also on the basis of the blueprint agenda of WHO. So I think we are at the point really where we are facing an enormous crisis. We're in the middle of a crisis, and we basically see how in the past we have, I wouldn't say we have failed, because we had fantastic results in the past, but we have not made optimal use of what happens in the scientific world and translating that into really tangible, let's say, antibodies, vaccines that you can use in to prevent or mitigate a large outbreak in humans and animals alike. I think that's the message for today. And I think in the afternoon session, I think we can discuss a number of these things, how these things can be speeded up. And I think I mentioned a couple of problems yesterday. Also, the difficulties we have in Europe with the use of laboratory animals, all the hurdles we have there. And I think the ethics there, it's clear, but we have to keep in mind that if you get big delays there, that's another problem to face. So what we may be doing this afternoon is really look at on the one hand, the achievements, but on the other hand, also, this is an example, and a very good example, money well spent, I would say, an example of how you can spend money on developing a system. And then what came out of the system, of course, there was different approaches there, and certain things were good, certain things were fantastic. And if I think, for instance, about the C1 system, if that is going to work at the end of the day, it's a pity the project doesn't run another 5 years, but basically we may be looking into the future there and see whether we can exploit these kind of achievements, where indeed, if it's true that we can at the end of the day, with little purification, make a 10-fold or a 30-fold recovery of antigens in a fungal system, for instance, I think that would be fantastic also for very rapidly spreading the things over the world. I was a firm believer in the bacillus system, as you can imagine, but now when I see the fungal system, the C1 system, I think I'm losing my belief a little bit and moving into another direction if it's going to work. So there's lessons to be learned, and that will continue in the future as well. So Jean-Christophe, I think this is more or less for myself. An overview.
Yes, there are a lot of lessons learned, and in fact, from the C1 assessment, something which was not also appreciated is that reaching these high yields also open the way to a much better purification, even in animal health, because you can afford a purification system that cannot work for lower yields. Also, the modular vaccine is, again, the best compromise to achieve the mass or the surge manufacturing capacity and to deliver globally the vaccines. And after the COVID-19 crisis, I think mRNA vaccines will also be around. It's on par. Maybe we'll see which system will be the easiest to manufacture and to handle. But the modular vaccine is also a very nice tool now for increasing the scientific knowledge, because it will help to dissect the immune response in a very fine way, because you play exactly with the particle, the subunit you put, the type of adjuvant, you can control everything, and then you can observe the results after immunization. So I think that will also be a basis for increasing the immunology world and vaccinology knowledge at large.
Yeah, and I think what was important also, discussions we had with Joris and others about let's say the example we have of the influenza vaccination in humans, where we also have a modular system where on a biannual basis we adapt the vaccine, and I think with the technologies as we have described them and as we have worked on, it will be quite feasible to do a similar thing in the future to rapidly replace the antigenic component of the vaccine, by a new strain that representing a new strain that popped up.
Yeah,
and I think we have a very good module there for
indeed, and we need that because we are still expecting the big one in terms of flu and the next pandemic, which did not occur with H1N1, and we know that we have to be extremely fast if we don't want to see again the same damages as today.
I think H1N1, you say, was not such a bad one. Still 500,000 people
but
died.
we are expecting it. But it's still to be expected.
I think that I see Jeff Almond waving his hand. So Jeff, could we give you the floor?
I'm sorry, I was muted. I was going to pick up on a couple of points you both made. To what extent has the world been revolutionized by RNA? Because, you know, I see the C1 results, they look very encouraging. We've got more options in E. coli. We can do the modular and the subunit building and so on. But, you know, the attraction of RNA suddenly is enormous. We've seen a response in manufacturing going from 0 to 2 billion doses, Mrna are claiming, and Pfizer, you know, for a single year. This supply chain for that is relatively straightforward. The single factory can produce many different vaccines just by a sequence change. We know that they're going to play with that to make, and I know Mrna are doing these things, to co-express 2 or 3 or 4 or 5 messenger RNAs in the same cell to allow assembly de novo of subviral particles or VLP type particles in response to the RNA vaccination. And I think we're also going to see modulation of RNA so that it is better able to stimulate cellular responses. Now, if we can drive the cost of RNA down and of course solve some of the problems with the stability, which I'm aware is already happening. It seems to me that becomes the platform for manufacture, which is almost unbeatable. Because any capacity that you have for any vaccine can suddenly be put onto the new vaccine because the process is going to be exactly the same. So I wonder to what extent you guys share the view that we've seen a revolution in vaccine technology through the application of the RNA.
If I may kick off, Jeff, I to a certain extent I agree with you. What we have seen for the messenger RNA vaccines is exemplary, but you also immediately mentioned the problems there. The versatility is fantastic, but first of all, we don't know anything about longevity of protection. We don't know; it's too early days. But that we may expect that will not be the major problem. The major problem I see is really cost on the one hand. I think cost is a major thing, and stability of the product. If you want to vaccinate in Africa, if you want to vaccinate Southeast Asia, if you want to vaccinate the world, it will be relatively difficult unless the cost issue and the stability issue would be solved. And I think there may be a place for both. Yeah, for the more classical techniques, if we call C1 a classical technique, or even the E. coli produced antigens, where you really go for antigen itself. But I agree. Yeah, I agree with you that we really have to look into that. That's progressing insights. Yeah.
Sorry, if I could just come back. I'm not saying RNA will replace everything. I don't think it's the answer for prokaryotic antigens, right? Because you're making them inside a eukaryotic cell with the mRNA approach, and quite often that's going to be a problem. So for prokaryotic protein vaccines or prokaryotic antigens, of course, for glycoconjugates, it's never going to work. So it doesn't cover the whole spectrum, but there are a lot, and particularly in the virology area, that I think it can address. And I think the costs will come down, because once you start making it 2 billion scale, the costs will fall dramatically. And I think also this, I'm hearing that the stability problems are also being solved. So we can expect, I'm sure, pretty stable vaccines at 4 degrees in liquid. And if not, then freeze dried. But I'm a little more bullish about their future, perhaps, and they may then overcome the need for a eukaryotic expression system like C1. I don't want to be negative on C1 because it looks really interesting, but the world has been revolutionized by this experience we've had in the last 12 months, it seems to me.
Yes, if I can comment on that, I think mRNA, yes, the COVID-19 is opening a new way, at least for human vaccines. I'm a bit skeptical on the capacity to stabilize and to have a cheap product, even if the manufacturing of RNA itself can be cheap. The lipid formulation is still complex, 4 to 5 different lipids. People have worked on formulation of RNA since the 90s and for siRNA, and still it's not really solved. And for animal health applications outside of pets, it's difficult to see how we could be used in the field conditions because you need multiviols and so on. And in the spirit of ZAPI, having a technology that can fulfill both human vaccines and animal vaccines, mRNA are not there. Maybe in the future, but again, also maybe a great scientific tool, but not for everything at this stage. Ronen?
Yeah, I don't want to challenge the great innovation of the RNA, but I want to comment on two aspects. First of all, we believe that we haven't tried yet, that we can also express more than one antigen in one cell line. So this is something that you can probably can apply also to C1 technology, maybe 2 or 3 even. In addition to that, I think once we have established the cell lines, we can also replace the antigen in a rapidly matter. For instance, we believe that in 2 months, if you need to have to develop a new antigen because of mutations and other variations, I think this is something that you can also do in C1. So in this respect, I think the advantage of the RNA is not that far from what we can show in the C1. Obviously now everybody thinks that the RNA seems to be a very good methodology, and I agree with that. By the way, I at the beginning thought that they actually printing the RNA, but they also produce it in E. coli. So I think that altogether we are talking about production, a live production system. So probably once I think if we continue and manage to establish the C1 as a production host firmly for antigen, I think the gap between the advantages between RNA and C1 will be narrowing. That's what I want to say.
Okay, Jeroen, you wanted your turn? Do you
yes,
want
I was just typing like crazy in the chat, but this is easier. I think, yeah, about the cost of the mRNA vaccines, I think we've learned from PCR. When PCR was first discovered and commercialized, we also thought that, oh, this is going to be much less expensive very, very soon, and that didn't happen. And I think we should also understand that there are certain limits to manufacturing of mRNAs, especially the larger ones, that we are very far from making that less expensive, and there are certainly limits that we need to take into account, and especially in the veterinary field, and especially again for developing countries. I don't see that approach for animals coming to market soon. Realistically, I think that the replicons, for example, the ones that are being developed at MSD, for example, based on VIV, Venereal encephalitis virus. I think that is a different story. So that's one of the interesting developments in my opinion. But the ones that are used now for humans based on mRNA, I don't see that happening soon for animals. But that's just my opinion. And also the type of immune response is of course quite different. If you administer mRNA or subunit with a certain adjuvant or a live generated vaccine, so it's not all the same thing that we are comparing here.
Mr. Peter Punt, do you have a comment? You're on mute. You're on mute.
And so I think indeed in the fungal systems, the increases can still be significant, because I mean, of course, it's an established system for producing fungal proteins, but for new proteins like vaccines or antibodies, it is developing. But I think the achievements in the C1 system are quite promising. I mean, and of course, I mean, all the registration and regulatory things need to be addressed, and that is a serious thing. But the flexibility, I think, is as what Ronan was saying, is clear.
Likan?
Yeah, I mean, just agreeing and echoing what is being said, I guess. And I know Jeff is not suggesting the other technologies not to be further developed, but I would agree with Jeff in the sense that the RNA, the validation of that technology by Mrna and Pfizer is a wake-up call that this new technology can work. So I guess we will begin to see a bit more of it in different, probably companies that are already working on it. And the advantage those companies had is because they have been developing different products in the RNA field for a while. So they have developed that technology, and they were able to quickly put that into practice when it was necessary. Yes, it is expensive, but I mean, the proof of it is in the efficacy, and I guess it's early days. I agree, it's early days. That is not to put to limit the potential of the C1, because in terms of the ability of the potential of the system to actually generate high yields of complex protein, it is great. But I do also agree, hopefully later today we can talk about the regulatory implications of a new platform, just like transients. What are the regulatory hurdles, and what do you need to do in so-called peacetime or in a pandemic situation? So that would be fantastic to discuss, really. But I think with all of this, the more new technology, the better. And we can't have just one system.
I fully agree with you, Likan. In fact, that was my comment. In my experience, relatively long now with new vaccine technologies, I'm not aware of a universal technology that can address everything. There are always limitations because, again, the combination does not fit for some reason. So eventually, we may need several systems to be ready to go for addressing new targets. Martin?
Only a comment about what was said. I think the game changer was not only the messenger RNA itself, because that's known for a long time. We have tested this for rabies, for H1N1, and it worked, but it was never allowed to use the lipids. So the game changer is that they are now efficiently packaged. And this will change everything for the future, also for the replicons, what Jeroen mentioned. I see there now a complete shift, because the argument 20 years was always it's not possible to use all these new lipids and all these transfection reagents, or however you want to call it. And now it's used in hundreds of millions of people. So I think if there is no adverse effects, this is now changing it completely. It's not the messenger RNA itself. The problem was always the delivery. And if that is solved now, I see that we will have a new dimension, and there is information that you can mix 5, 8, I think the highest number is 10 different messenger RNAs. So you have also not anymore the problem of having several targets in one vaccine. And I would dare to look in the future, and maybe we will see vaccines of 5, 6, or 7 different antigens mixed by RNAs. And the only risk I see is there any long-term effects? Because that can nobody tell at the moment, because we inject a lot of RNA, and the reactivities, if you look into the papers, is really interesting. It's a very high reactivity, even in people older than 80 years, which normally you don't see. So there is something going on with a very, very good immune response, even in the elderly. So the question for me is more, what do we know in 10 years? But it's, for me, it's a really the biggest game changer, and it's especially the packaging, because this opens a completely new field for all RNAs, as Sharon mentioned, all the replicons.
Yeah, I think it will be interesting to test these new vaccines when we will be available in target species to see if they work in the same way. And just a comment, indeed, I noticed, because it's very clear in phase 3 trials, high level of reactiveness, but probably it's part of the success of why the RNA vaccines are working, because you induce a danger signal that typically you don't want to have, because we have safer and more and more pure vaccines, and to the point that they are less efficient. So that's a system and a knowledge we have very well in animal health. So maybe we come back also with mRNA vaccines to all the school type of vaccine where you need to have a strong danger signal to be efficient.
I agree that we're in early days, but what I'm hearing are, concomitant administration of RNAs that are designed to alter the proteasome, to modulate the interferon response and the danger signal, to assembly subparticles to specifically trigger a cellular immune response as well as an antibody response, to co-administer messenger RNAs for some of the cytokines that can lead to if you like, a refinement of the immune response that you get. So it is early days, but I do think we, as Martin said, we're at the start of a revolution here that could get really very, very exciting for vaccine design and administration. I absolutely agree that we're not there yet with the costs and the stability, but it's such a young technology, and those things are almost certain to be refined as we move ahead. I know we had this sort of revolution with DNA 20 or 30 years ago, and it came to nothing. So we have to be cautious.
Yes, I know that very well. I worked 10 years on DNA for a very good tool for science, but not for a product.
So did we, and of course it didn't go anywhere. But I think the RNA is a different story because of the fact that it's astonishing what we've seen with 95% efficacy for Mrna and Pfizer. And the other thing that has been announced, Mrna was asked, what does it take if they have to make a new serotype COVID vaccine to deal with, let's say, the Brazilian strain, if it really was antigenically resistant? 5 weeks is the answer. In other words, you change the sequence, you continue the factory in the same way, you grow up your E. coli plasmid, and you go. And really, the licensure of a variant would be quite easy just on immunogenicity, small trial. And Ab, to your point, I think this is going to revolutionize flu vaccine responses for the future. So pandemic preparedness, they'll be in the driving seat for future flu pandemics.
But in principle, this is not a prerogative for mRNA, because also with the other techniques that we have to date, even the C1, there's also a matter of weeks that you can change the formula, obviously. And the other point you made about co-administering messengers or any substance that will induce cytokine responses, I would be a little bit reluctant to do that, given the possibility might induce autoimmune responses. But I think there will be a major regulatory hurdle there.
You're probably right, but as I say, it's early days and it's an exciting new technology.
Absolutely.
So I have now Joris and Peter Punt, who wants to make a comment first.
Yeah, so I was thinking, I mean, is there not also, in a way, a hybrid situation that will occur for rapid responses? I can imagine you can spend the costs of RNA vaccines, which, well, I agree with what was discussed, that whether the prices would go down there, I don't know, but that you would use for the common vaccinations, like for flu. But I mean, of course, for COVID, we will get also a common vaccination required, because it's now all over the place. So every year there would be probably now also COVID vaccinations, that you would use then the protein vaccines or the vector vaccines for that, so that there will be a hybrid situation that will remain, because you want to be able to have a fast response. And so it's not a one size fits all there.
Okay, Joris?
Yes, thank you, Jean-Christophe. The point started by Jeff is, of course, totally right, and it's a big lesson. That being said, I would like to see it from another perspective. As you said, Jean-Christophe, I would not put all my eggs in the same basket, because our long life in biology, vaccinology learns that we did not see one example where one technology fits all. But that being said, of course, the RNA story is extremely interesting. But what does it learn? And I would like to come back how it generated so quickly. So I have seen recently a long interview with Moncef Slaoui, and he explains there very clearly the genesis of this RAP project in the US. They looked what was available in February last year. And thanks to all the public investments, there were a lot of results on messenger RNA. But in addition to that, on vector vaccines, and in addition to that, 2 types of Novavax vaccines. And that's where they decided each of them gets more or less $2 billion to go forward for the sake of protection of the people. So what I like to say, we have to put all this in perspective, and it is the same with the technologies developed by ZAPI. If we come at a point where we are known and where it is available, then there is investment ready for that. However, at the European side, and Aap mentioned, and we should insist on that, on some basic problems we face here in Europe, for example, for animal trials and aversion to innovation, which made that on the other side of the Atlantic, we have a more favorable environment to say, yes, go for it. But that being said, this technology, like the C1 we have developed here, and the ZAPI multimodal approach, is as interesting, at least to my feeling. But that being said, messenger RNA is not only a technically interesting project, it will of course evolve and will be more accessible. I'm convinced of that, but let the time play. But the same is true for the ZAPI project. Thank you.